and Chrissy for having us today. As Bea said, I'm Heather. I'm at Newcastle University, and today I'm going to be talking about the work that we've been doing with Nova, who is who I collaborate with on this project, but also do lots of field work with, explore the rocky shores of the UK and wider with, but it isn't just the two of us. There's also Mike, who I think is here in the audience with us, and Steve and other people who go out and explore these shores with us. And we collect data all over the country and really use these data to investigate climate-driven changes on our benthic communities and in the regional seas around the UK. Okay, this is driving now. So the project that we're going to be talking about is called MARCLIM. That is the time series that we're going to use, but then we're going to go into a bit more detail about how we use those data. It's a UK-wide project, and we use it to synthesize and interpret both historical data and current data that we've collected for intertidal rocky shore species around the UK. And we're repeating classic broad-scale surveys of the 1930s and the 50s, and we've taken that forward into today, starting from 2002 all the way forward to now. And Nova's been out this week, I think, collecting data on the shore in the sunshine that we're having now. And that's established a long-term network of sites to monitor any change that we're seeing on those shores. We collect data on 87 species of algae and invertebrate, and they act as indicator species, allowing us to track the shifts and changes in that community structure that we see. We all know that our climate is changing, and we know that in recent years they've been recorded as some of the hottest on record, and that leads to extreme events that we see and that are becoming more prevalent in nature. So we can track the responses of any changes in our intertidal communities using these indicator species as part of the time series. Ultimately, we can use these data to inform marine policy and make recommendations for the management of our systems and address the implications that this might have for our society. So you can see here this map of the UK where we might go out and about collecting these data. We have over 100 sites that we visit regularly, but we might have more sites as well. This is very much dependent upon funding. As you all know, funding is a challenge, especially when it comes to time series data. So we very much do things on a shoestring, and where we can get more funding, we will go and visit more sites. So you can see here, this is Steve Hawkins collecting data back in the 80s, I think this is, and Nova and I collecting data more recently. There's a large body of work that we've been developing around this theme, but today we're going to focus very much on the field surveys that we've been doing, the types of field experiments and lab experiments that have been informed by our field surveys. We're going to bring in a little bit of microclimate data that we've been collecting that feeds into these field and lab experiments that we've been doing, how this feeds into mapping and mechanistic species distribution models, and then how this really feeds into managing and thinking about our marine protected areas using these time series data. So we all know that marine and coastal ecosystems are spatially and temporally variable, but they're also experiencing these unprecedented changes in response to global anthropogenic forcing of those atmospheric processes, coupled with additional human activities. We know that they're operating at global, regional, and local scales, and we know that sustained observations from biota across multiple trophic levels and across broad geographical scales are required to really separate those climate-driven signals from the noise of those natural fluctuations, and that we can use this to really disentangle the responses of species and communities and ecosystems to global drivers from regional and local scale impacts. Time series are really important to think about to be a primary resource to enable us to track the status and the functioning of those ecosystems in responses to lots of these changes that we're seeing. The time series that we've been collecting under MARCLIM have shown some of the fastest biogeographic range limits for any species responses to this climate change that we're seeing. So we're seeing poleward extensions of northern boundaries for some of our Lusitanian gastropods, so Phorcus lineatus and Steromphala umbilicalis, for our macroalgae bifurcaria here, and for our barnacles perforatus. But we're also seeing retractions of some of those boreal species, so the barnacle semibalanus balanoides and the Kelpalaria esculenta. They're retracting in their range edges. We can track those species responses for the warming and cooling that we're seeing in the UK climate change. So the types of temperatures that we're seeing over time is really being reflected in the movements of those species. The rates of those change are species specific, so with resultant changes to the biodiversity and ecosystem functioning. And the long-term impact of those extreme weather events can also be tracked by the time series that we're seeing. If you have a look at this graph here, you can see that that change in the mean abundance is consistent over time. So we can see that we're seeing these increases in species abundances, especially for those warm water species, over time consistently. And you can see that at every site that we've looked at, this is just an example for North Wales and the southwest of England, that the temperatures are also increasing over time. And you can see that these patterns are reflected across France and across the southwest of England. So we're seeing consistent increases in these species as we look across these areas. The responses that we're seeing are very species specific though. So when we're thinking about that ecosystem structure, we really have to think about how individual species are responding to the changes that we're seeing in the temperatures that we're recording. So you can see here on this graph, for example, this is the time series from a place called Cellar Beach in the southwest, and this has got annual records since 1952. The data that we're seeing here show a tight relationship to local sea surface temperature, and you can see in red the abundances of the cathamelid barnacles, and in blue the abundances of the boreal barnacles, semibalanus balanoides. As we get the warmer years, we see an increase in our Lusitanian cathamelid barnacles, and as we get colder years, we see an increase in abundance of our boreal barnacles, semibalanus balanoides. So we can track the abundance of these species in response to the changes in the sea surface temperatures that we're experiencing. Using similar data collected for a range of invertebrate species, we can also start to think about predicting the probability of the presence of species through dynamic species distribution models. So this is taking into account the temporal aspects of a species distribution, including predictability of future abundance categories and the expected persistence times of local populations, and how these might respond to different environmental or anthropogenic drivers. So you can see here there's an example for two of our top shell species. These are our Lusitanian top shell species, so they're responding to warmer temperatures. And this is based on 9 years worth of field data. So the abundances were recorded on the categorical scale. That's what we use when we're doing our field surveys. And then we can have a look at those abundance categories against mean sea surface temperatures in the winter and the wave fetch as explanatory variables. And we can start to think about predicting where they might be in the future and how they might respond to different climate changes in response to their current distributions. So start to have a prediction against where they might be in the future and how they might persist into that future. And we can then start to make predictions on that in terms of a management perspective as well. I'm now going to hand over to Nova to think about the pervasive element of this in terms of extreme events.
Yeah, thanks everybody and welcome. Not only does Malcolm look at long-term pervasive climate change impacts, but we can also, because we're surveying every site all the way around several regional seas of the UK every year, we can also look at the impact of extreme events as well. And I'm not going to talk about heatwave events here. I'm actually going to look at the extreme storm event that happened in the winter of 2013-14. You probably all remember it. It was unmatched in intensity for over half a century. There were the largest wave heights recorded, the highest rainfall, but interestingly, there were no extreme temperature events. So climate-wise, it was pretty similar to the long-term average for winter. What we did see, though, because we have so many different sites around the place, is we saw different effects happening on every single shore. The pictures on the left here, so the pictures that show our lovely colleague, Professor Hawkins, who, as Mike Burrows will know, is always bending over and flashing his backside, and if he is, I take a picture of it. But he's actually doing serious fieldwork here. And before the storm event, as you can see at Travone, there was a huge mussel bed carpeting the lower shore. When we went back immediately after the storm event, almost the entire bed of mussels and all the associated barnacles and limpets had been ripped off and disappeared as well. Whereas moving ever so slightly up the coast of Crackington Haven on the right-hand side with the pictures, just a few miles up, you can see here something very different happened. The top picture shows our site at the end of the cliff on what used to be a sandy beach, whereas immediately after the storm, all the sand was removed from the bay and there was new rock surface uncovered. So what we saw then was colonisation of foundational species. And we noticed that every site we went to, we saw very, very different impacts, sometimes biological, sometimes physical. And it just goes to show that you can't just predict what's going to happen, even at adjacent shores from the same event. And we've also set up experiments. These experiments, again, mirror experiments that Mike Burrows did way, way back in time for his PhD. And we thought it'd be very interesting to go and reset these experiments up now that climate has got warmer, and to see whether the competition between the warm water Lusitanian species, so the camelids, and the boreal colder water species, semi-balanced balanoides, and the invasive species Ostrominis vedestus, whether they were changing in terms of competitive settlement and dominance compared to when Mike first went and looked at them. And again, we saw that there was a real effect of climate change, because the Lusitanian warm water species are doing far better these days, but also because semi-balanced balanoides is a gregarious settler, it needs to look for adult individuals in order to be able to settle nearby them. Because there are so few of these around in the southwest now, it's actually compounding the issue for semi-balance, and we're seeing a real decline in the abundance, to the extent that sometimes when I go on a southwest shore, I really struggle to find any semi-balance at all. And we also do a lot of experiments in both laboratories to look at multiple stresses. So not just sea surface temperature, but also looking at ocean acidification as well. And I'm going to hand back over to Heather for a short while she explains these to you.
So what we're trying to do here is take what we're seeing in the field and actually bring some of those back into the laboratory. So we know that the organisms and the algae that we're looking at in the field are experiencing multiple stressors in the field. We know that isn't just temperature that they're experiencing, it's actually affecting what's happening. We know that there's lots of things that are going on out there. So what we're trying to do here is understand the drivers that are impacting on that change that we're seeing. So we've set up a mesocosm here. This one is actually based in Newcastle in the Dublin Marine Laboratory. There's a comparable system that Nova will show you in a minute that's based down at the MBA. And we're trying to have a look at those drivers in respect to the different regional seas that we have in the UK, because we know that we're seeing temperature gradients across the west coast, but we also know that we're seeing temperature gradients between the east and the west coast. So we've set up these systems so that we can actually run mesocosm experiments to change some of those multiple stresses and actually look at the different species that we have and look at the drivers for change on those different species. So as Nova says, change the temperature, change things like the ocean acidification, introduce different nutrients, for example, and actually see whether we can bring in multiple stressor experiments under mesocosm systems, and then look at that in the real world and field environments to look at the comparison between the two. And Nova's just going to tell you a little bit about that in terms of biogeographical challenges.
Yep, so the reason that we started doing these experiments is that we found that a lot of species distribution models, they currently rely on correlating species distributions and quite coarse scale climatic data. These are okay, but they're not particularly accurate. So we wanted to start moving beyond using these correlative SDMs and really look at the organism level responses that are driving these large scale range shifts. Because what's actually happening is biological mechanisms inside each individual organism, wherever they are in the species distributional range, are responding to climate change, sometimes in different ways. So if we can start to look at niche tracking in multivariate space, it's going to give us a much better mechanistic understanding, which means that we then start to measure physiological performance. We look at environmental sensitivities, how different populations acclimate and adapt to different ones. And also, I think one thing that's really important, and a lot of time people forget this, is species aren't existing in the wild in isolation. So we need to also remember that not just to run experiments in the lab with species in isolation as well, but we also need to look at biological interactions between them.
And so one of the things that we wanted to build into this was actually looking at those microhabitats and those niches in the real world and in the environment and on the shore. And so we started to deploy what are called robo-limpets. And a colleague of ours in Portugal, Fernando Lima, developed these robo-limpets. And these provide temperature data that mimics the internal body temperature of intertidal mollusks. And so we deployed these in out on natural open rock with a robolimpet, but also a logger that records the temperature on the rock surface itself. We deployed them in shady areas and in pools. So in those niches where these organisms actually live, the limpets and many of the other invertebrates that we track, they record hourly temperatures on both the rocks and representing the internal temperature of these organisms. This allows us to gather that contextual information for those intertidal species and the sites where we have long-term time series data. So you can see that we have loggers out in all of these areas around the UK. We also have them in some areas around Europe as well. The sites in green are actually funded by Natural England, whereas the other sites we funded out with some of the research projects. And we've paired them together. So we have an understanding of the rock surface temperature and an understanding of the internal body temperature of the organisms. This is actually allowing us to get really high resolution temperature data for those areas, which is of a higher resolution than we would have from that sea surface temperature data that covers a much greater area that's often used to predict those scenarios. And then we can then take those data to inform some of those experiments that we've just been talking about in those mesocosms. Just to give you an idea, and this is a very quick look, so we started to deploy these in 2018, so we've now got about 5 or 6 years worth of data, and we are starting to see that, you know, in the open rocks we're getting much lower lows than we would in some of those niche habitats, but we haven't really looked at these data properly yet. This is sort of the next step to really pull some of these fine scale data into the experiments that we've been doing. And Nova's just going to give you an example of some of these experiments.
Yeah, so here's a quick schematic of some of the multiple stressor experiments that we've done already. We can raise or lower the water temperature, the air temperature, as Heather said, look at ocean acidification impacts as well. And one thing we found in response to multiple stressors was that we actually found the shell shape changing from some gastropods as well, which was quite interesting, because this is especially when you see where they seal to the rock, and if their shell shape and their aperture is changing, this means that they're not actually able to seal themselves particularly well when the tide's out, and therefore this might also cause additional effects that we wouldn't necessarily notice where we're just looking at abundance and distribution. We're also very fortunate to have friends at the University of Oxford who ran transcriptomics on our samples as well and identified several different genes that are upregulated or downregulated in response to warming, cooling, and ocean acidification as well. So this is starting to give us a much more in-depth idea of exactly how all of these different species are responding to climate change, and may help us understand why then the different biogeographic range shifts are happening. Here's just a quick example of one of the experiments, just to show you how we set them out. We look at different temperatures, different pH levels, and we can also do things like look at the effect of microplastics in a more harmful warming acidic world as well. And we're fortunate enough to have younger people than us who are eager to join the team and to develop new techniques as well. So this is one of the PhD students who's co-funded by the MBA based at the University of Liverpool, Dina. Dina is looking at eDNA and working out whether this is an effective method to look for Markram species in the water column. She's come with me to several locations, including quite a lot of sites that we have up in North Scotland, and she's testing all of these techniques to see just how effective eDNA is as a detection tool versus our eyes. And you can see from the results here that they're actually not as good, thankfully, or else Heather, Mike and I wouldn't have a job anymore, not as good as visual surveys to detect species at the species level. They start at the genus level to become reasonably similar, and at the family level, they're actually pretty good and better than us at detecting family level ones. And if you look at the bar graph on the left-hand side, this also shows you nicely as well how the left-hand bar in Scotland has got an awful lot more boreal species present, whereas as you move through the bars south towards southwest England, you can see that there's a lot more in terms of Lusitanian and cosmopolitan species as well. So we do think that this technique has got promise.
And just sticking on the theme of eDNA, we are also developing a technique based on subtidal benthic communities. And this was established in 2018. So not using the time series from Markland, but established time series in subtidal communities. Looking at how we can develop species-specific primers for eDNA to identify marine invasive species across this area here of the northeast and just tipping over the border there into Scotland. So we use a technique where we submit settlement panels into the marine environment, and we leave them in from about January to September. And once a month, we will remove one settlement panel from the water. And then we will use those settlement panels that we've taken out of the water to test our species-specific primers to see if we can pick up the presence of an invasive species on those panels before we can visually see that species. So we've developed species-specific primers for Ostrominius modestus and Corella yumyota, and we are in the process of developing a third species-specific primer for Scheißerparella japonica. And then we've tested 3 additional primers for the invasive species for 3 additional species in this area. And what we found is that we can pick up the signature for invasive species before we can visually identify them. But these species did then grow into these communities once those panels were left in the water for a period of time. So these sites are located very close to the time series sites. So we know that there's a potential for these species to come into that area. We know that we can then think about using this as a rapid response mechanism. We can build this into our management and conservation protocols, but we can also start to think about how we can pick these up in the time series and know that these species might be coming into those areas of the time series from adjacent areas in that way. So we're using the eDNA in a slightly different way in this respect because it's the species-specific eDNA, but we know that we're getting some good results here. So again, showing promise, but we do recognise that there are limitations in this area. Thinking about that management and conservation train of thought, that brings us quite nicely onto how we're using these long-term time series and thinking about marine protected areas. We obviously have a lot of data, and it can be used for more than just tracking those changes in species abundances and thinking about how our communities are changing. We obviously have some of these sites within marine protected areas and some outside of marine protected areas. So it offers a unique opportunity to think about how our marine protected areas might be being impacted by some of these long-term pervasive changes. Marine protected areas are often assessed by things like condition assessments. So what we can do is use the time series to think about how we might assess the condition of a marine protected area. So an example of this we can see here with the Heartland Point to Tyntagle Marine Conservation Zone. We have communities of species at all of these different sites within the Marine Conservation Zone. So we can see how those communities are changing across space and across time. And then we can assess the features of a particular element of that marine conservation zone and the attributes associated with that to see whether that marine conservation zone is hitting its target and whether the particular targets of that feature are changing over time or not. So we can really use these data in an applied way to think about the conservation of these areas and how that management might be targeted in a slightly different way. And I'll just hand over to Nova to think about the rest of that management.
Yeah. So one thing that we've been involved in since the very start of the MSIP process is writing the report cards, and now we've transitioned into the rolling evidence base to now providing information on the coastal intertidal habitats for MSIP as well. And I think this is a really important output to policymakers as well, because I think it's a really nice visual guide with some really clear messages and identifying the key challenges, which means that we can get our message out to people that wouldn't necessarily hear about the science in a way that's accessible, and hopefully will make them realise that we still need to have regular time series funding so we can actually track, monitor and project into the future exactly what's going to happen in our UK seas. And we've just been doing some work with CFAS as well on the UK marine warming pathways. Again, we were looking at different scenarios. We were pulling in data, including the Markland time series, to then look at not only site-specific changes, but then scale this up from a local to the global level, and then to start to characterise the pathways as well. And hopefully we'll get a second phase of this in 2025, which means we can carry on working with them and utilising the data even more. We've developed indicators of good environmental status First of all, for the Marine Strategy Framework Directive, when we were part of Europe, and then again for the UK Marine Strategy, no, we're not. And we have different things that we do. So here we enter Mike Burrows, stage left. Mike has done a huge amount of really insightful statistical analysis and modelling. First of all, he calculated the species thermal index for every single marklim species. Then what he did is he was able to then develop a community thermal index from these data. And they showed some quite interesting things. So for the invertebrates, there was a very, very different relationship with sea surface temperature to the macroalgal species in the time series. If you look at the middle panels, there was also a different response in the southwest of Britain compared to what's happening up in Shetland. And we're also able to then plot this community thermal index, CTI, on maps so that people can see exactly how the different species are contributing to different thermal indices depending on where you are. And no surprise, the further north that you go in the UK, the lower the community thermal index is. And we're also being involved in larger projects, including this international one. So we're developing an essential ocean variable for macroalgae as part of Goose. Because we've got a fantastic time series for macroalgae at lots of different sites, I've been able to trial the standard operational procedures for the essential ocean variable and compare them against the marklim data. Methods are also being trialled by Jarrett Burns in the US at subtidal sites to see if we can do diving surveys as well. And hopefully at the forthcoming International Temperate Reef Symposium, we will be at one of the workshops and liaising with other people working globally on macroalgae to start to bring all of these ideas together.
And that's us. It's obviously funded by lots of different organisations, and we work with a huge array of different people in order to achieve everything that we've presented there. Thank you very much.
So thank you so much for giving me the opportunity to present this work today, Bea, and the convener of the forum really at large. I'm based at University of Edinburgh as a university teacher, but my research is mainly focused on understanding the impacts of human activities on deep sea benthic ecosystems. And as Bea just mentioned, I had the pleasure of co-leading the working group looking at the effects of climate change on Scottish deep seas. We've just finished our work as a working group, and we've produced 2 outputs, a MASTS story map and a policy brief that I'll show you today, and I hope that you'll find these interesting and be able to use them in your own work. Very briefly, I'll go over the reason why we thought this working group was needed. I'll mention all the members of the working group because this was really a team effort. We'll really quickly look over our policy, our story map, and our policy brief. I'll give you some of our key take-home messages, and I just want to, right at the end, very quickly highlight the opportunity that MASTS working groups really offer. So there's other working groups that are already formed within MASTS, but I just would like to say that it's a really good way of getting some tangible outputs produced in a quite short time period. So if you have some things you've been meaning to get done, but didn't quite know how to get them done, I think the MASTS working group might be the way forward. So stay tuned if you're maybe interested in that aspect of MASTS as well. I probably don't have to present this in very much detail in this forum, but of course, we know that our oceans have changed and the deep sea as part of our oceans is changing because of human activities, including emissions of greenhouse gases. So that the ocean, of course, plays an important buffer in our climate system, and so it's taking on lots of additional heat, but also taking up anthropogenic CO2. And that has created change in both ocean currents, in biology, and the chemistry of our seawater. I think you're much better equipped than me to understand all of these changes. In the deep sea, and we really define the deep sea as any water column or seabed below 200 meters depth, we try and summarize the main impacts of climate change and ocean acidification across 4 main drivers. So warming, acidification, deoxygenation, and changes in particulate organic carbon fluxes, so the food that comes down to the seabed. And you will have seen potentially this figure before, or a variation of it. Is usually how we try and summarize the activities, but also the impacts of climate change and ocean acidification on the deep sea. When we look at a bit more details, if we look at the projections for end of century conditions in the deep sea, we expect that average temperatures will increase in excess of 1 degree, that we'll see a decrease in pH and greater than 0.3 units, a decrease in dissolved oxygen concentration of up to 3.7%, with a lot of regional variability here and expansion in some regions of oxygen minimum zones. And again, a decrease in the amount of food that comes down to the seafloor of about 40 to 55%, so a really significant decrease. And while we are slowly, as deep sea ecologists, trying to make sense of what these drivers will, or the changes that these drivers will lead to in terms of deep sea species and ecosystems, I think it's clear that at large, the royal we, society, and the general public sees the deep sea as this kind of remote, potentially unimpacted by human activities area, with very limited impact on our daily lives. And of course, this is not true. The deep sea delivers a huge range of ecosystem services and products to us. And so we really need to be, as scientists, better at communicating what are the impacts of climate change on deep sea ecosystems and why should we care. So you might have seen this comic before. It's used quite a bit in the deep sea world, at least. And it dates back to 1983, but it's actually still pretty relevant, unfortunately. And this is really the background that gave us the idea of the MASTS working group looking at the effects of climate change on the Scottish deep sea. I just want to take a moment to really highlight that this was a working group, so there was a really big element of team effort, and that was very much a teamwork makes the dream work kind of approach to all of this. So Christina Beck, a colleague from University of Edinburgh, was co-PI with me. Colin Moffat, Erin Kettle, Dominic Anderson, Daniel De Jong, Teresa Fernandez, Lucy Goodwin, Kelly James, Andrew Sweetman, and Emily Haig were all working group members. And we had additional support from Bhavani Nawaswamy and Chrissy Oni. But I do want to highlight a few key contributions further. So Emily Haig was our technical developer for the story map specifically, and she managed to complete her PhD and become a postdoc and do this story map all in a few months, 6 months that we had her on the working group. So we really wouldn't have a tangible tool that we can then now use and showcase for our work without Emily. So a huge thank you to her. Chrissy One supported the working group throughout all the stages and had the difficult task of trying to corral this group of scientists to deliver the outputs that we promised we would. So that's never an easy task, we all know that. And also designed the policy brief and helped us with the dissemination over the last few weeks. So a massive thank you to Chrissy. And then we also had external reviewers looking over our story map, making sure that the messages we were conveying were clear and correct. And so a really special thank you to Bhavani, Christine, and John, who gave us loads of really, really helpful feedback on our story map. So just wanted to highlight that this was very much a team effort, even though I'm the one presenting to the forum today. Okay, so you've heard me kind of throwing around this word story map. So let's actually dig into what a story map is and what we have produced really. So we have two outputs. This story map, which is, if you're not familiar with ArcGIS and ArcGIS kind of programs that we use to create map, a story map is a online tool that we can use to create interactive reports, basically, that showcase a whole range of data, whether that is maps or text. You can embed videos, images. You can create little interactive sliders around your images. And it basically creates a website that you can then use to showcase a whole range of information. So we use the story map infrastructure to showcase what we know and what we understand currently of the effects of climate change on the Scottish deep sea. And I'll show you very shortly what it looks like. And then accompanying the story map, we decided to also make a policy brief, which was much shorter and to the point. And so I guess you're getting the sense here that we're really trying to target two audiences here. The story map is very much dedicated to try and communicate this research to the general public, whereas the policy brief is much more intended for policymakers. So it's much shorter and to the point. I think the best way to actually show you the story map is to go to the story map. So I'll just exit my presentation for a moment. And I'm hoping that you still see my screen. Please do shout if that's not the case. But this is basically what the story map looks like. You'll get the link sent in the chat very shortly. If not already, I'll do it just after I finish my talk. So it's basically a website that you can navigate to the same way you would any website. So you'd scroll down. The story map is organized in different sections. So we have an introduction, a section on the environmental effects of climate change, looking at the stressors that I highlighted to you at the beginning of my talk. And then we have a series of case studies looking at sediments, sponges, corals, demersal fish. We've also included a conclusion section where we have a summary of the information that you find in the story map, as well as some recommendations. And as you scroll through, we've tried to come up with some text that is understandable for a broad range of members of the general public. We've tried to limit the use of jargon, and that sometimes was the most difficult part of the working group's efforts, was to try and agree on what constitutes jargon. It sounds like a very easy task, but actually we kind of seem to forget that we use technical terms in our everyday as scientists. And then we try and include tools that a reader can interact with. So RGS is particularly, or story maps, sorry, are particularly well suited for these types of maps. You can navigate through to different regions to know more about specific areas of the deep sea in Scotland and get a whole range of information on these offshore marine regions, as we call them here. We haven't reinvented the wheel, so a lot of the information that is here comes from other sources. So we've embedded used videos that we found from other researchers within the MASTS community or from, further abroad, which are really good videos and that we could use straight off the back, or good figures that we thought were particularly meaningful. Another feature that's really helpful in story maps is that you can also embed images and make these images kind of quite interactive. So for example, in the sediment case study, we used a slider tool to try and engage the reader a little bit more and make them realize that actually sediment regions are full of really cool animals and really interesting organisms like the xenophyophores. And so you can use these sliders to reveal to the reader where the different species are on the picture, for example. And that can be a really nice way to highlight the diversity that exists in the deep sea and their importance to the reader. You might have noticed that in our text, we've tried and make everything or relate the references of the things that, of the elements that we showcase. So we've tried to embed links to the literature where we find our specific piece of information, for example. So there are references throughout used in the story map that you can, so you can see exactly where we get a specific part of or key bit of information that we highlight in our story map.